Hello students. Today's topic is urine formation. The formation of urine is the result of the process like glomerular filtration, reabsorption and secretion. These processes occur in different parts of the nephron. Now I am going to discuss about each part of the nephron where these different processes are carried on. So let's start with glomerular filtration. Glomerular filtration is the first step of urine formation. It is the filtration of blood. It is carried out in the glomerulus. Thus, it is known as glomerulus filtration. Now, this glomerular filtration takes place in the kidney. The kidney nearly filter about 1100 to 1200 milliliter of blood per minute, which constitute roughly 1/5 of the blood pumped by each of our ventricle of the heart in a minute. Now, this glomerular capillary blood pressure causes filtration of the blood through three layers. The layers are the endothelium of the glomerular blood vessels, the epithelium of the Bowman's capsule, and the basement membrane present between the above two layers that I have mentioned just now. Now, there are some podocyte cells. These are the epithelial cells of the Bowman's capsule. These are arranged in such a manner that some minute spaces called filtration slits or slit pores occur between them. On account of high pressure in the glomerular capillary, the substances are filtered through these pores into the lumen of the Bowman's capsule. But WBC, RBC and plasma protein have high molecular weight, so they are unable to pass through this filter. That's why this process of filtration is known as ultrafiltration. Now here I am going to tell you a very important term that is called glomerular filtration rate. Now it is the amount of filtrate formed by the kidney per minute. In a healthy adult person, it is about 125 milliliter per minute, that is 180 liter per day. The glomerular filtration rate is regulated by one of the efficient mechanism carried out by the JGA, that is juxtaglomerular apparatus. Now, what is this juxtaglomerular apparatus? It is a special sensitive region that is formed by the cellular modifications in the distal convoluted tubule and the afferent arteriole at the location of their contact. Now, this aperture includes three things. What are they? They are the granular juxtaglomerular cells in the afferent arteriole, the macula densa cells of the distal convoluted tubule, and the agranular lattice cells situated in between the above two cells. Now, what happens if the GFR, that is the glomerular filtration rate, falls? When this GFR falls, it can activate the JG cells, that is the juxtaglomerular cells, to release renin, that is a hormone, which can stimulate the glomerular blood flow and thereby the GFR come back to normal state. So, this is very important to remember. The next step in the urine formation is the selective reabsorption. So, what is selective reabsorption? Let's discuss. Selective reabsorption is the second step in the formation of urine from the filtrate. Now, the urine released is only 1.5 liter as compared to the volume of the filtrate that is formed per day. Just now, I told you that 180 liter of filtrate are produced in a day, that is within 24 hours. But do we ever excrete 180 liter of urine? No, not that. We only excrete 1.5 liter of urine throughout the day. So, it suggests that as much as 99% of the material in the filtrate is reabsorbed by the renal tubule. Thus, this process is called selective reabsorption or simply reabsorption. So, depending upon the type of molecules that has been absorbed or that will be absorbed, the movement into and out of the epithelial cell in different segments of the nephron occur either by passive transport or by active transport. So, here you have to learn how these molecules are reabsorbed. Like water and urea are reabsorbed by passive transport. That is, water is reabsorbed by osmosis and urea is absorbed by simple diffusion. Next is glucose and amino acid. These are reabsorbed by active transport. And the reabsorption of sodium ion occurs both by passive transport as well as by active transport. Now comes the third step of the urine formation, that is the tubular secretion. It is also an important step in urine formation. Certain chemicals in the blood that are not removed by filtration from the glomerular capillaries are removed by this process of tubular secretion. Now, it helps in the maintenance of ionic and acid-base balance of the body fluid by removing the chemicals like foreign bodies and certain ions. So here you will learn about the function of the tubules. Now here you may think that what are the differences between tubular reabsorption and tubular secretion? Look, tubular reabsorption involves the absorption of water and useful solutes from the glomerular filtrate into the blood. But tubular secretion is the passage of waste material from the blood into the filtrate of the nephron. So this is just vice versa. Tubular reabsorption occurs by back diffusion and active transport, while tubular secretion takes place only by active transport. And one more very important difference is that tubular reabsorption only take place or mostly take place in the proximal convoluted tubule, while the tubular secretion takes place in the distal convoluted tubule. So these are few of the differences that you may understand. Now you see, in tubular reabsorption, this type of reabsorption does not take place in animal that lack glomerulus, that is in desert animal or marine fishes. So dear students, this was all about the 3 steps of the urine formation. Hope you got some idea about the formation of urine. In the next video, I will be discussing about the counter current mechanism. Thanks for watching this video.